Hey everyone, and welcome. Today we're talking about Benedict's test. It's this wonderfully simple experiment that reveals the hidden world of sugars, and it is seriously cool. So let's just jump right in. Okay, so picture this. You've got a clear liquid. Could be juice, a sample from a biology lab, pretty much anything. And you need to know if it has a certain type of sugar hiding inside. How on earth are you supposed to know just by looking at it? Well, the answer is pretty incredible. You do it with a rainbow and a test tube. And today, we're going to break down exactly how this little piece of lab magic works. All right, first things first. Let's get into the secret chemical reaction that makes this whole thing possible. Seriously, this is where the science gets really, really cool. So this awesome, colorful experiment, it's called Benedict's test. It's a total classic in chemistry and biology labs for finding what are called reducing sugars. But it's not just for school labs. It has some major real-world uses, from checking the sugar in our food to, believe it or not, its historical use in urine tests to help screen for diabetes.
But wait, what is a reducing sugar? Think of it like this. It's a type of sugar with a special superpower. It's super generous. It has the ability to just give away one of its electrons to another chemical. And that generosity, that ability to donate an electron, is the key to this entire jest.
And this is where it all clicks into place. See, the Benedict solution is packed with these blue-colored copper ions. When a reducing sugar shows up and you add a little heat, that generous sugar donates an electron straight to one of those copper ions. And that one simple act, that electron donation, it changes everything. Okay, this transformation is absolutely the key to the whole thing. Before the reaction, you got these blue copper-2 ions, and they're just dissolved in the water, totally invisible. But once they grab that electron from the sugar, poof, they turn into red copper-1 oxide. And this new stuff, it's not soluble. It forms a solid, a precipitate, that crashes out of the solution. And that is what causes that dramatic color change we see. So you're probably wondering, how can I make this rainbow happen? Well, let's get to it and assemble our virtual lab kit. First up, the magic potion itself, Benedict's reagent. It's actually made of 3 key things. You've got copper sulfate, that's what gives us those all-important blue copper ions. Then you have sodium carbonate and sodium citrate, and they basically team up to create the perfect alkaline conditions for the reaction to actually happen. And of course, you can't do the experiment without a few other essentials. You obviously need your sample, the liquid you want to test, some clean test tubes, and a dropper for measuring. And this is a big one. You need heat. A water bath is perfect. Seriously, without heat, nothing's going to happen. Okay, we've got the science down, we've got our materials ready, so now it's time for the main event, the 5 simple steps to make the magic happen. Let's walk through this together, step by step. First, you're going to take 2 milliliters of your sample and pop it in a clean test tube. Step 2, add the same amount of Benedict's reagent. Easy. Step 3, give it a little swirl to mix it up. Step 4 is the action part. Stick that test tube in a boiling water bath for about 2 to 5 minutes. And finally, step 5, take it out, let it cool, and see what you've got. It's honestly that simple. And here we are, the moment of truth. Once that test tube has cooled down, it's time to decode what it's telling us by looking at that amazing color spectrum. You know, this is what makes the test so brilliant. It's not just a yes or no answer. It's what we call semi-quantitative. If your solution stays blue, that means nope, no reducing sugars here. But if it turns green, you've got a tiny trace. Yellow means there's a low amount, orange tells you there's a moderate concentration, and if you get that deep brick red color, bam, that's a high concentration. You are literally seeing the concentration of sugar right there in the tube. All right, let's do a quick fire recap of the key takeaways here. 1, Benedict's test, it's all about finding reducing sugars. 2, to make it work, you need 2 things, the reagent and heat. 3, any color change away from that original blue means it's a positive result. And 4, and this is the best part, that final color tells you just how much sugar you're dealing with. I mean, how cool is that? A simple mix of chemicals and a little bit of heat lets us see something that was totally invisible just a few minutes before. It really makes you wonder, right? What else is out there, totally invisible to us, that science can help us see, if we just know where to look?